Now we're going to skim through the type 2 hypersensitivity reaction or hypersensitivity mechanism and examples. Type 2 hypersensitivity reactions involve antibody-mediated destruction of cells. It is also known as cytotoxic reaction. In this hypersensitivity reaction, a specific antibody IgG or IgM bound to cell surface antigen and destroy the cell. If the cell is microorganism, killing of cell is beneficial to host. However, type 2 hypersensitivity, the cells are own RBC, so that's kind of dangerous. And the killing of cell can occur by one of the three mechanisms. They are complement mediated cell lysis, antibody dependent cell mediated cytotoxicity or ADCC, then opsonization. So you could see a picture for this infected cell lysis, phagocytosis. Some other mechanisms are also happening, which we will be learning. So complement mediated lysis of cell. Complement system is a system of lytic enzymes that are usually inactive in blood. Enzymes of complement system are activated by antigen antibody complex. When antibody binds to antigen microorganisms or RBCs, they form AgAb complex. AgAb complex can activate complement system by three different mechanisms: classical pathway, alternate pathway, and lectin pathway. Activated complement proceeds in cascade mechanism. When complement is activated on the surface of cell RBC, it causes lysis of cell. Antibody dependent cell mediated cytotoxicity or ADCC. Antibody dependent cellular cytotoxicity. Antibody binds with antigen by its Fab portion. However, Fc region of the antibody has receptor on cytotoxic cells. So antibody cross-link target cells, microorganisms or RBC with cytotoxic cells and promote killing. Then most cytotoxic cells contain storage of hydrolytic and digestive enzymes. These enzymes are released on the surface of target cell MOs and RBs of or target cells, killing them. Here antibody itself does not kill or destroy cell, but rather mediate killing by presenting antigen to cytotoxic cell. Similarly, cytotoxic cell depends upon antibody to bind antigen. So this mechanism is known as antibody dependent cell mediated cytotoxicity. Then opsonization. When antigen enters into host body, antibodies are produced. Antibody binds to antigen through Fab region. Fc region of antibody remains free. Then phagocytic cells such as neutrophils, macrophages, and monocytes have receptors that can bind to Fc region of antibody. The receptor is known as FCR. In this case, antibody molecule directly cross-links antigen, microorganism or RBC or target cell with phagocytic cells. This cross-linkage, antibody binds to antigen through Fab region. Fc region of antibody remains free. Phagocytic cells such as neutrophils, macrophages, and monocytes have receptors that can bind to Fc region of antibody. The receptor is known as FCR. In this case, antibody molecule directly cross-links antigen, microorganism or RBC or antigen cells with phagocytic cells. This cross-linkage activates phagocytic cells and increases the rate of phagocytosis. This increased rate of phagocytosis by binding of antibody to antigen is called opsonization. Examples: blood transfusion reaction. ABO blood transfusion reaction is an example of type 2 hypersensitivity reaction. Human RBC contains A or B antigen as a major antigen in the surface of RBC. Other minor antigens such as RH, Cal, Duffy, etc. are also present. Antibodies to ABO antigen is called isohemagglutinin and usually IgM class, whereas antibodies or to other minor antigens are of IgG class. An individual with blood group A recognizes B antigen like epitope blood group B as foreign and produces isohemagglutinin antibodies. The same individual does not produce antibodies to A antigen as it is similar to self-antigen, so that state of tolerance exists. If individual with blood group A is transfused with the blood containing B antigen, then transfusion reaction occurs in which anti-B isohemagglutinin antibodies binds with B blood cells and mediate destruction of transfused RBC by complement activation. Since lysis of RBC occurs in intravascular space, free hemoglobin appears in urine. Hemoglobin can be converted into bilirubin, which is highly toxic to tissues. For treatment of this hypersensitivity reaction, transfusion should be stopped immediately. Blood transfusion reaction occurs immediately in case of mismatched ABO antigen. In this case, transfused RBCs are lysed by complement. In case of minor antigen mismatch, delayed reaction occurs, and in this case, transfused RBCs are lysed by opsonization, IgG antibody, and no free hemoglobin appears in urine. Hemolytic disease of newborn erythroblastosis fetalis. Hemolytic disease of newborn develops when maternal IgG antibody specific fetal blood group crosses placenta and destroys fetal RBC. The consequences of transfer of antibody can be minor, serious, or lethal or to fetus. Serious hemolytic disease of newborn develops when Rh negative mother conceive Rh positive fetus, which cause erythroblastosis fetalis. During pregnancy, fetal RBCs are separated from other circulation by a layer of cell called trophoblast. During her first pregnancy with Rh positive fetus, mother circulation is not exposed to enough fetal RBC to activate Rh specific B cells for antibody production. At the time of delivery, large amount of fetal umbilical cord blood enters to mother circulation. This fetal blood activates mother Rh specific B cells, resulting in production of plasma cell and memory cell. The plasma cell produce IgM antibodies which binds and destroy fetal RBCs from mother circulation, but the memory cell remains which treat any subsequent pregnancy with Rh positive fetus. Activation of memory cells in subsequent pregnancy with Rh positive fetus cause production of IgG antibodies which can cross placenta and destroy fetal RBC. Mild to severe anemia develops in fetus and sometimes fatal. The conversion of hemoglobin to bilirubin produces additional threat to newborn because bilirubin may accumulate in brain and damage it. This hemolytic disease of newborn can be prevented by injecting preformed antibodies against Rh antigen to mother at around 28 weeks of pregnancy and within 48 hours of first antibodies marked as ROG. These antibodies bind to RBCs of fetus and mother circulation and clear before B cell activation. Then drug-induced hemolytic anemia. Certain drugs such as penicillin, cephalosporin, and streptomycin can absorb non-specifically to protein on surface to RBCs forming complex similar to hapten carrier complex. In some patients, these complex induce formation of antibodies which binds to drugs on RBC and induce complement-mediated lysis of RBC and thus produce progressive anemia. This drug-induced hemolytic anemia is an example of type 2 hypersensitivity reaction. Type 1 hypersensitivity reaction in detail is the immediate type of reaction mediated by IgE. This is called anaphylactic reaction or allergy. Now we are talking about type 1. It is induced by certain types of antigens called allergens such as pollen grains, dandruff, dusts, food components, etc. Allergens induce humoral antibody response by the same mechanism as other soluble antigens in the generation of antibody secreting plasma cells and memory cells, but the difference is that the plasma cells secrete IgE class of antibody. Allergens include humoral immunity response by the same mechanism as other soluble antigens in the generation of antibody secreting plasma cells and memory cells, but the difference is that the plasma cells secrete IgE class of antibody. This IgE class of antibodies have high binding affinity to Fc receptor on the surface of tissue mast cells and blood basophils. Mast cells and basophils bound by IgE are said to be sensitized. A later exposure to the same allergens cross-links the membrane-bound IgE on sensitized mast cell and basophils, causing degranulation of these cells. Degranulation causes release of pharmacologically active mediators which act on surrounding tissue, causing vasodilation and smooth muscle contraction, which may be either systemic or localized. This effect is also termed allergy. So degranulation causes release of pharmacological active mediators which act on surrounding tissue causing vasodilation and smooth muscle contraction, which may be either systemic or localized. This effect is also termed as allergy. Mechanism of type 1 hypersensitivity reaction. Mechanism of type 1 hypersensitivity reaction. Development of allergy or anaphylactic reaction requires two doses of antigen. First dose is known as sensitizing dose and second dose is known as shocking dose. Production of IgE antibody. When an antigen or allergen enters into host, production of antibody begins. At first, allergen is processed and presented by antigen presenting cells to CD4 T cells. Activated CD4 T cells divide to form T helper cells and memory cell. T helper cells produce IL-4. At the same time, B cell bind to antigen and presence of IPC and APC and IL-4 and gets activated. So activated B cells divide to form plasma cells and memory cells. Up to this step, mechanism is similar to that of normal immune response. The difference between type 1 hypersensitivity and normal immune response is that the plasma cell produces IgE antibody instead of IgM or IgG. Then sensitization. Fc region of IgE antibody has receptor on the surface of tissue mast cells and blood basophils. So the IgE antibody binds to FcR1 of mast cells and basophils. Next, sensitization. Fc region of IgE antibody has receptor on the surface of tissue mast cells and blood basophils. So the IgE antibody binds to FcR1 of mast cells and basophils. This binding of IgE to mast cell and basophil is known as sensitization, and the mast cells and basophils are said to be sensitized. Then shocking dose of antigen when the same antigen enter into the same host. For second time in life, antigen cross-linked with the Fab region of IgE molecule on the surface of mast cell or basophil. Degranulation of mast cell. The cross-linking of antigen or allergen to IgE antibody causes degradation of mast cell and basophils, releases various pharmacologically active chemicals such as histamine, heparin, serotonin, cytokine, leukotriene, prostaglandin, etc. Anaphylactic reaction. These active chemical mediators act on surrounding tissue, producing various symptoms of allergy such as vasodilation, smooth muscle contraction, mucus production, sneezing, etc. The allergic reaction may be localized or systemic depending upon types of allergy. Mechanism of activation and degranulation of mast cell. The intracellular signaling events that ultimately results in mast cell degranulation are manifested involving cooperation among various protein and lipid kinase and phosphatase and rearrangement of cytoskeleton. The biochemical events in mast cell activation and degranulation can be divided into three steps: phosphorylation, methylation, and adylation. Adenylation. Phosphorylation. Binding of H allergen on FcER1 bound antibody activates protein tyrosine kinase PTK, then phosphorylates phospholipase C, which converts phosphatidylinositol 4,5-biphosphate TAP2 into diacylglycerol and inositol triphosphate IP3. And IP3 is a potent mobilizer which causes pore formation and release Ca++ ions. DAG activate protein kinase C, PKC. Methylation. Cross-linkage of FcRn also activates an enzyme that converts phosphatidylserine into phosphatidylenolamine. Eventually, PE is methylated to form phosphatidylcholine by the enzyme phospholipid methyltransferase 1 and 2, PMT1 and PMT2. The accumulation of PC on exterior surface of plasma membrane causes an increased membrane fluidity and facilitates the formation of Ca++ channel and Ca2+ chain ions enter through the channel. Then Ca++ ion and PTK activates mitogen activated protein kinase MAP kinase. MAP kinase activates phospholipase A2, which promotes breakdown of phosphatidylcholine into lyso-PC and arachidonic acid. Arachidonic acid is converted into potent mediator leukotrienes and prostaglandin D2. Activated MAP kinase also induces secretion of cytokines by increasing transcription of cytokine genes. Then adenylation. Fc epsilon R1 cross-linkage also activates the membrane adenylate cyclase enzyme, leading to transient increase of CAMP within 15 seconds. Acylated drop in which CAMP is mediated by protein kinase and is required for degranulation of mast cells. CAMP-dependent protein kinase phosphorylates the granule membrane protein, thereby changing the permeability of the granule to water and Ca++ ions, causing swelling of granules. Consequently, swelling of granules and formation of soluble N-ethylamide attachment receptor snare protein complex facilitate fusion of membrane and release of pharmacological chemical products, which is responsible for allergic symptoms. Clinical manifestation of type 1 hypersensitivity reaction. Clinical symptoms ranges from life-threatening conditions such as systemic anaphylaxis and severe asthma to localized reactions such as hay fever and eczema. Systemic anaphylaxis, it is a shock-like and often fatal state which is usually initiated by allergen directly into bloodstream or absorbed from gut or skin. The symptoms are labored breathing, drop in blood pressure, smooth muscle contraction, bronchial constriction, suffocation. Localized hypersensitivity, it is limited to specific targeted tissue. Wait a minute. First one was systemic anaphylaxis. It's a shock-like and often fatal state with labored breathing, drop in blood pressure, smooth muscle contraction, bronchial constriction, then suffocation. Localized hypersensitivity, it is limited to specific target tissue or organs, often involving epithelial surface at the site of allergen entry. The tendency to manifest hypersensitivity reaction is inherited and is called atopy. Atopic allergy induces wide range of IgE-mediated disorders such as hay fever, asthma, food allergy, atopic dermatitis, or eczema. Type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction factors causing immune complex formation, mechanism and types. The reaction of antibody with antigen regenerates immune complex. In most of the cases, these immune complexes are removed from blood circulation. Some immune complexes are removed by phagocytic action of phagocytic cells in blood. Most other immune complexes are first carried by blood to spleen where they are destroyed by macrophages. Complement system is also needed for removal of immune complexes from blood to spleen. In some cases, large amount of immune complexes are formed and deposited on various body parts and leads to tissue damage resulting in type 3 hypersensitivity reaction. If immune complexes are not removed from blood, they accumulate on wall of blood vessels and on tissue where filtration of blood and plasma occurs such as glomerular membrane, synovial membrane of joints, etc. Then type 3 hypersensitivity reaction is characterized by deposition of immune complexes on various tissues such as wall of blood vessels, glomerular basement membrane of kidney, synovial membrane of joints, and choroidal axis of brain. Deposition of immune complexes initiates reaction resulting in damage of surrounding tissue and cause inflammation. Factors that cause deposition of immune complex and increase susceptibility to type 3 hypersensitivity reactions. Persistent infection. The persistent infections such as malaria, large number of immune complexes are formed and deposited in tissues. Then complement deficiency. Complement removes immune complexes from blood, but when complement system is deficient, large amount of immune complexes circulates in blood and deposits in tissues. Persistent infection, then complement deficiency. Complement removes tissue immune complexes from blood, but when complement system is deficient, large amount of immune complexes circulate in blood and deposit in tissues. Then autoimmunity. In autoimmune disease, large amount of immune complexes are formed and deposited in tissues. Autoimmunity. In autoimmune disease, large amount of immune complexes are formed and deposited in tissue. Then genetic defects. In certain genetic defects, small and soluble immune complexes are formed that cannot be phagocytosed. Mechanism of type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction develops when immune complex activates C3A and C5A components of complement system. C3A and C5A are lymphotoxin, anaphylatoxin that causes localized mast cell degranulation. Degranulation of mast cell releases histamine, which increases vascular permeability of blood capillaries. This facilitates deposition of immune complexes on wall of blood vessels. C5A, C3A, and C5B, C7 also acts as chemotactic factors for neutrophils. So it attracts neutrophils at the site of immune complex deposition. Then C3b acts as opsonin by binding with immune complex. Neutrophil binds to C3b coated immune complex by means of type 1 complement receptor, which is specific for C3b. The neutrophils attempts to phagocytose the immune complex, but phagocytosis is not possible because immune complexes are deposited on basement membrane. So the neutrophil release lytic enzymes to destroy immune complex. Then the lytic enzymes cause tissue damage surrounding of immune complex deposits, resulting hypersensitivity reaction. Furthermore, complement protein can also contribute to tissue destruction. Then types of type 3 hypersensitivity reaction. Localized type 3 hypersensitivity reaction. Acute arthritis reaction. Martha's reaction is an example of localized type 3 hypersensitivity reaction. When antigen is injected or enters intradermally or subcutaneously, they bind with antibody to form localized immune complex which mediate acute arthritis reactions within 4 to 8 hours. As the reaction develops, localized tissue damage and vascular damage results in accumulation of fluid edema and RBC erythema at the site of antigen entry. The severity of reaction can vary from mild swelling and redness to tissue necrosis. And when antigen is injected or enters intradermally or subcutaneously, they bind with antibody to form localized immune complexes which mediate acute arthritis reaction within 4 to 8 hours. After the reaction develops, localized tissue damage and vascular damage results in accumulation of fluids and RBC, erythema, at the site of antigen entry. The severity of reaction can vary from mild swelling and redness to tissue necrosis. Generalized type 3 hypersensitivity reaction. Serum sickness is an example of generalized type 3 hypersensitivity reaction. When large amount of antigen enter blood serum and bind to antibody, circulating immune complexes forms. If antigens are insignificantly excess compared to antibody, the immune complexes formed are smaller and soluble, which are not phagocytosed by phagocytic cells, leading to type 3 hypersensitivity reaction. The manifestation of serum sickness depends on the quantity of immune complex as well as the overall site of deposition. The site may vary, but accumulation of complexes occur at site of blood filtration. Generalized type 3 hypersensitivity reaction at different sites result in different diseases such as glomerulonephritis, vasculitis, arthritis, synovial joints. Type 3 hypersensitivity reaction, factors causing immune complex formation, mechanism and types. Next, last one is the type 4 hypersensitivity reaction or delayed type hypersensitivity. Type 4 hypersensitivity reaction is also known as delayed type hypersensitivity. When some subpopulation of activated T helper cells encounters certain antigen, they secrete cytokines. When some subpopulation of type 4 hypersensitivity reaction is also known as delayed type hypersensitivity. When some subpopulation of activated T helper cells encounters certain antigen, they secrete cytokines that induce a localized inflammatory reaction called delayed hypersensitivity or DTH. The reaction is characterized by influxes of non-specific inflammatory cell, particularly macrophages. So DTH occurs slowly and reaches a peak level within 48 to 74 hours after second encounter of antigen. In DTH, tissue damage is mediated by TH cells and macrophages, but not by antibodies. Although DTH causes some tissue damage, it is very important defense mechanism against intracellular microorganisms such as Mycobacterium tuberculosis, Mycobacterium leprae, and Brucella sp, etc. Sensitizing phase of DTH. Sensitization phase, intracellular bacteria or PPC, CD4 plus TH, TH1 cells, generally DTH mediated cells, TH1 cells, generally CD8 plus cells, occasionally antigen presenting cells, macrophages, Langerhans cells. DTH begins with initial sensitization by primary contact with antigen. At first, antigen is processed and presented by antigen presenting cells to CD4 plus T cells. CD4 plus T cells are activated to form TH cells. CD4 plus T cells are activated to form TH cells. During this, TH cells are clonally expanded by binding with MHC to molecule carrying antigen by appropriate APCs. Varieties of APCs have been shown to be involved in activation of DTH response, including Langerhans cells and macrophages. CD4 plus T cells are the primary cell activated during synthesizing phase of DTH response. However, in some cases, CD4 plus cells are also found to induce DTH response. Effective phase of DTH. A subsequent or a second time exposure to antigen induces the effective phase. In this phase, TH1 cell secretes varieties of cytokines that recruits and activates macrophages and other non-specific inflammatory cells to the site of antigen injection. Macrophages are the principal effector of DTH response. The activated macrophages exhibit increased level of phagocytosis and increased ability to kill the antigen. microorganisms by various cytotoxic lytic enzymes. Activated macrophages releases lytic enzymes that damage surrounding tissues and intracellular bacteria. The influx and activation of macrophages in DTH is important in host defense against intracellular bacteria and parasites, where circulating antibodies cannot reach them. Activated macrophages release lytic enzymes that damage surrounding tissues and intracellular bacteria. The influx and activation of macrophages in DTH is important in host defense against intracellular bacteria and parasites, where circulating antibodies cannot reach them. Increased phagocytic activity and built-up lytic enzyme from macrophages in the area of infection leads to non-specific destruction of tissues and intracellular pathogens. Generally, pathogens are killed rapidly with little tissue damage. However, in some cases, especially if the antigen is not easily cleared, a prolonged DTH response can itself become destructive to host as intensive and chronic inflammation develops into a visible granulomatous reaction. A granuloma develops when continuous activation of macrophages induces the epithelioid shape and sometimes fusion of macrophages to form multinucleated giant cells. These giant cells displace normal cells, forming palpable nodules and release high concentration of lytic enzymes which destroy surrounding tissue leading to necrosis. Examples of type 4 hypersensitivity reaction: tuberculin skin test, lepromin test, brucellogen test.